AP Biology students, let's go over the second section in Unit 2. And this section is going to be about eukaryotic cells. Remember that eukaryotic cells are larger, they're more complex, and they have something called a membrane-bound organelle, while prokaryotes don't have a nucleus, are smaller, and are less complex. I always like to try to throw in evolution and how these things evolve throughout time when talking about the different processes within biology. Basically, what happened for the nucleus and other membrane-bound organelles to evolve is that infolding occurred and it wrapped itself around the DNA of a cell. And this happened some 2 billion years ago. And this was a huge advantage for cells because you could compartmentalize and protect the DNA compared to other cells without a nucleus. Now, this infolding and the production of a nucleus occurred before something called the endosymbiotic theory, which talks about how the chloroplast and mitochondria may have evolved. This endosymbiotic theory is basically how mitochondria and chloroplast evolved within our eukaryotic cells. What happened was billions of years ago, a eukaryotic cell engulfed an aerobic bacteria and a cyanobacteria, and instead of destroying them or eating them, they kept them around. This was a huge evolutionary advantage because this cyanobacteria would later evolve into modern-day chloroplast, while this aerobic bacteria would evolve into today's mitochondria. Let's quickly go over the timeline of how eukaryotic cells evolved. Ancestral prokaryotic cells billions of years ago had infolding with its membrane, and this infolding created the nucleus by wrapping a membrane around the DNA. Later on, some of the cells took on aerobic bacteria, which later became the mitochondria, and these aerobic bacteria were able to break down sugars really easily and produce a lot of energy. Other cells took on the cyanobacteria, which were able to take in sunlight and inorganic compounds and produce sugar. So what happened was this endosymbiosis is responsible for creating today's mitochondria and chloroplasts in our current eukaryotic cells. Yes, I know it sounds crazy to think about that our cells pretty much have bacteria floating around in them, but over the next couple slides, I'm going to cover some of the evidence that supports this endosymbiotic theory. The first piece of evidence for the endosymbiotic theory is DNA. Inside the mitochondria, you have circular DNA called mitochondrial DNA. And if you remember from back when we talked about prokaryotic cells have circular DNA. Only prokaryotic cells have circular DNA, so this is some of the evidence that supports that mitochondria were once living, free-floating bacteria that had their own separate DNA. So yes, not all the DNA in a cell is located in the nucleus. The mitochondria has its own circular DNA. One of the best pieces of evidence for the endosymbiotic theory is the membranes that we find in mitochondria. There are two membranes. The outermost membrane, you can see here in red, resembles that of a eukaryotic cell's membrane. The inner membrane, seen here in green, resembles that of a prokaryotic cell's membrane. And that should make sense to you because the eukaryotic cell that enveloped these billions of years ago would have wrapped its membrane and been the outermost portion membrane here when looking at these double membranes. Second, there are ribosomes within the mitochondria. And if you remember, having ribosomes is a requirement for cells. You have to be able to produce proteins. And not only do they have ribosomes, but these ribosomes also resemble the ribosomes of prokaryotes. Prokaryotic cells and eukaryotic cells have different types of ribosomes, and the ones that we find in mitochondria resemble those of prokaryotes. Next, mitochondria reproduce very similarly to the binary fission we see in bacteria and other prokaryotes. They divide and they split whenever they need to have more of these mitochondria in a eukaryotic cell. So their reproduction pattern looks like that of a bacteria cell rather than a eukaryotic cell. And lastly, this is kind of not the greatest piece of evidence, but it's still evidence. The size, they are similar to the size of bacteria cells. You can see this is what a mitochondria looks like. This is what a bacteria looks like. You can see the DNA there and the membranes here. Yes, these mitochondria are about the same size of a bacteria cell. I like showing this phylogenetic tree when talking about the endosymbiotic theory so we can see the time frame that these would have occurred. You can see that mitochondria would have been enveloped before the splitting of animals, fungi, and plants, but chloroplasts would have been enveloped only on the plant's lineage since we don't see them in animals or fungi. One of the most notable structures when talking about eukaryotic cells is obviously the nucleus. And this is a circular structure found near the center of a cell, and its function is to contain and protect the DNA. Now, a lot of times we think of a nucleus that looks like this. You can see the outer membrane here. There's actually two membranes. You have the chromatin here in the middle and something called the nucleolus. I like to show this image when talking about the nucleus because most of the time the DNA is going to be not condensed, and it kind of looks like a bowl of spaghetti, as you can see here. Only a short period of a cell's life is going to have the DNA in this compact X structure. So most of the time when we look at cells, this DNA is uncondensed. It's not packed up into a tight X pattern. It's in this unpacked chromatin form. Now, the nucleolus isn't really a separate whole organelle. It's part of the nucleus, but it's a dark region within the nucleus that creates ribosomes, or at least parts of the ribosome. And lastly, the outside of the nucleus is called the nuclear envelope. And this is a double membrane that forms around the DNA. There are also things called nuclear pores. You can see these pores around the nucleus, and these are just basically holes that the mRNA is going to travel through for transcription and translation to occur. I absolutely love this meme. This is where I would put my nucleus, prokaryotic cells, if I had one, because they don't have one. Great meme.